Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on our screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Christina Morris from BioPharmaSpec.
Hi and welcome to our talk on host cell protein mass spectrometric analysis, where we're specifically discussing some of the strategies to consider in your process development toolkit. So just a quick overview of the points of discussion. We'll briefly describe why HCPs should be studied, the tools we have available and their benefits, and then I'll focus on key topics which are important to think about when planning the HCP analysis methodology and what you can expect to get out of the results. So I'll zip through this, as I'm sure most people in the audience are aware of the importance of studying host cell contaminants. HCPs are endogenous proteins that co-purify with the host cell during biopharmaceutical drug production. The purification procedures that are used are often multi-step processes that polish most but not all of the impurities out of the final drug product. And some of these HCPs may be immunogenic and or could lower drug efficacy. So it's important to assess what and how much of these residuals are present in your drug product and to be aware of how levels are changing throughout the purification, especially if any changes have been made to any stage of this process, which can change the HCP population. Manufacturers need to determine which HCPs to monitor as a critical quality attribute and which analytical methods are best, and to be aware that safety limits are product specific and defined on an individual basis. Although it has been suggested that for injectables, levels between 1 to 100 ppm may be acceptable. And finally, it's recommended that you monitor HCP numbers throughout the purification process in order to show clearance of the impurities and better understand your control strategies, which will allow you to make improvements to minimize the levels of HCPs. So advantages of MS-based analyses compared to other technologies include the high sensitivity, reproducibility, and specificity, and the ability to identify de novo a wide population of individual HCPs without the need for the specialized antibody reagents or product and process-specific method development. It's important to reiterate that LC-MS is not a replacement for the HCP ELISA test, but can provide an increased breadth of HCP identification as an orthogonal method. So MS-based quantitation strategies can be split into two main groups, those that use labeling technologies and those that are label-free. Labeling methods include techniques with multiple approaches within them, but they're broadly consisting of two categories, post-harvest or metabolically active labeling techniques. Post-harvest tagging strategies introduce labels at the protein or peptide level after cells have been lyzed, whereas the metabolic labeling involves endogenous incorporation of stable isotope labels in active cells. These labeling techniques can tag peptides chemically, metabolically, or enzymatically, and have been primarily developed for monitoring differential protein expression. A few examples of these techniques have been given in the table to the right. So labeling methods provide greater accuracy for quantitating proteins of interest, but increasingly label-free approaches are employed to replace or complement labeling methods due to the ease of experimental setup and the relatively low cost. Label-free approaches typically identify a larger number of proteins with a wider dynamic range of detection. They're less expensive because no special media or expensive reagents are required, and the workflows are simpler and faster as there's no need to determine the labeling efficiency. And because in HCP analysis we're quantitating very low abundance proteins, targeted mass spectrometric techniques such as multiple reaction monitoring or SWAS have produced the best results for these types of studies, which is why they're frequently used in HCP-MS. So we wanted to center this discussion on some of the options and considerations that might be helpful to think about after you've made the decision to perform MS-HCP analysis as part of your process development. As I've just mentioned, there are multiple ways to monitor proteins of interest using MS, multiple quantitation strategies and instruments, some of which are especially tailored for HCP measurement. Over the years at BPS, we've adopted specific approaches that we favor in order to maximize efficiency and optimize our detection capabilities for a specific client project. And within these approaches, 3 areas that we think warrant the extra attention, and these include the spectral or protein library, the samples to select for the quantitative analysis, and the data quality or expectations based on the MS technique and the processing. The workflow at the bottom of this slide illustrates a typical HCP analysis pathway for a Cyrex or Waters MS method to show where the 3 key areas come into play. In general, most HCP strategies will use a typical proteomics approach to assess the HCP profile in the sample, where proteins are digested into peptides and these act as models of an intact host cell protein. So reduction, alkylation, and digestion of all proteins within the mixture is performed, followed by online LC separation, usually reverse phase, and then mass spectrometric identification, quantitation using a high-res instrument like a quadruple time of flight mass spectrometer. HCP identification takes place through screening the sample data against a protein library that is specific to the host cell line, and information-dependent runs provide this data by cycling between MS survey scans and targeted MS-MS fragmentation of specific precursor signals, and then using selected standard spikes at known concentrations for the relative quantitation. The sample analysis is performed to identify and then estimate the mole values of the observed HCP areas. And this is done using data-independent acquisition methods like SWOF. And then finally, bioinformatics tools such as Progenesis or PLGS or the SWOF acquisition microapp are then used to perform the protein searches and reduce the false positives. So lastly, depending on your specific process development strategy and following the data review, there is the option to use a higher accuracy quantitative approach, for example, with the synthetic heavy labeled peptides coupled often with a triple quadrupole or an Orbitrap instrument. As we've seen in the previous slide, the spectral library is the first step to building your HCP quantitation strategy. IDA or DDA MS data collects information about the HCPs with high confidence, and the fragment ions observed in a dataset confirm the identity of an HCP peptide through amino acid sequence information. Bioinformatics searches match predicted peptide sequences in your data against sequences in a species-specific database of the host cell and assign a confidence score for the match. This step, as you can imagine, can be critical to what HCPs you may identify in your samples, and there are a few options when it comes to selecting the type of library to use, each with its own pros and cons that we've listed and that I'll go through. So more often than not, a library is made from a host cell lysate, which is likely to have host cell proteins that are present in high concentrations. This is especially useful for analysis, as although an IDA or DDA method provides more confident results, the sensitivity is not as great as the runs for quantitation, and the precursor ion selection is usually in the order of the most intense signals for MS fragmentation. So additionally, unlike pre-existing host cell line standards, the spectral library will be specific to the purification process for your drug product, which is particularly important as the smallest changes in the manufacturing process can result in changes to the HCP population. The downside is that the lysate will need to be generated, and this is not always straightforward. Specialist equipment or methods may be required that are not normally part of the process that you're familiar with, and this is especially true in cases where the active pharmaceutical is secreted from the host cell. So due to the complexity of a typical lysate, there is also the added cost associated with the library generation in order to ensure good LC separation and detection of the low abundance HCPs. Another option is the use of in-process and/or drug substance and final drug product samples for the basis of your library. This can be useful in cases where in-depth analysis of low abundance HCPs is not necessary and an overview is preferred, or perhaps a lysate is too difficult to generate. An advantage is that the sample preparation stages can include both the library prep and the HCP quantitation, which can significantly cut the analysis-related costs and the project timeline. And lastly, a spectral library can be generated using off-the-shelf host cell standards, which provides the advantage of having greater depth as you're including proteins from the whole proteome. In cases where there is a selection of different pre-existing host cell libraries, it will also reduce the expenditure associated with the setup of the library. But the downside is that it's not going to be process-specific. So following library generation, sample analysis takes place, Here, the DIA runs generate reproducible data sets that pick out very weak signals, which is needed when performing searches in samples further down the processing stream where you'd expect to see low-intensity HCPs. Deciding which samples are appropriate for quantitation will be specific to your aims, and we have demonstrated 3 strategies with different sample selection rationale that might assist in the decision-making. So an overview analysis where 1 or 2 process stages can be selected. For these types of strategies, perhaps you're not yet happy with certain stages of your process. You want comparison with an existing ELISA and to focus on a very specific stage to monitor improvement. So that might involve quantitating multiple batches or lots to check for any changes, say, throughout the year and to monitor the clearance. Alternatively, you have low HCP numbers validated with ELISA and you're happy with your upstream process steps, but you want an orthogonal assessment of downstream stages. So you can focus on just the drug product or the drug substance. Then we have a full assessment scenario using samples from multiple purification stages, all the way from upstream in process samples down to the drug product. And here you might not have a reliable ELISA, therefore there is a heavy reliance on other available orthogonal techniques. Or you may have changed an upstream processing step, risking changes in your HCP population, and your custom process specific ELISA needs to be redeveloped, pushing time constraints. Therefore, you can use MS to ensure quickly that there are no changes or new issues and that good clearance has been achieved. Or perhaps you're at the final stages of your product development and wanting to move forward with regulatory approval, and showing clearance is an important box to tick. And then lastly, a more accurate quantitative assessment using drug product sample may be appropriate for cases where there are specific HCPs of concern, perhaps at higher than expected levels, and you'd like a more accurate method of assessment using heavy label analogs. In theory, this tends to be performed on the final drug product, but if there are other stages of interest, these can also be analysed. Or maybe your total ppm estimates are on the threshold for your target and or you're unsure about a previously used MS relative quantitation method, and you want better validation of your final drug product. So the final stage of the analysis is the data review, and we want to highlight what expectations to bear in mind once you have produced your HCP results. It's important to know how the nature of the methods employed are linked to the data output, and there are 2 factors in particular that we feel should be noted when you're drawing conclusions about your sample data, and these are the software limitations and the LC-MS method setup. The data processing by proteomic software is required to provide the HCP matches against the spectral library and quantitate the large data set, usually in ppm micromoles per mole or micrograms per gram. Bioinformatics tools filter the data sets, which are usually composed of many replicates, and depending on the types of samples analysed, you can be presented with thousands of possible HCP hits. So the algorithms that sort through these mountains of spectra are fundamental to the HCP-MS strategy in terms of time efficiency. But as with any method, there are limitations that can lead to HCP misassignment or false positive identification. And these include underestimating peptide confidence scores, leading to a protein being excluded from a data set altogether, although this is often due to weak library data. Retention time alignment issues where areas for the same signal are wrongly assigned as different HCPs. Peak picking errors, especially for low abundance HCPs. So here the selection of the wrong area within the retention time window will lead to high method error across replicates. So for these reasons, we highly recommend manual review of selected HCPs. It won't catch everything, in part because of the associated challenges of attempting to create and interpret data over a 5 order of magnitude sensitivity range. However, by targeting, for example, the highest intensity hits, it is possible to weed out further any false identifications that can have a significant impact on total HCP estimates. And to conclude this discussion, we wanted to emphasize also that the LC-MS method that has been selected for your analysis will dictate the data quality. And a few examples include how many technical repeats have been used. A higher number can improve statistical confidence and ultimately provide further evidence for low abundance HCPs, which, as you can imagine, will assist with the manual review process. Whether you have supplemented the base library, so the protein library, with sample IDA-MS runs can make a difference to account for any HCPs missing from the library that are specific specifically found in your sample. And then the limitations are that some HCPs might be suppressed altogether as a result of dealing with the very high drug product in comparison to contaminants. So the LC method or the spike standards can make a difference here. And for example, 2D LCMS might be more useful in cases where there are specific low abundance HCPs of concern. So targeted relative quantitation has an advantage in terms of cost and ease of use, but the ionization and signal responses of the spike standards peptides will be different to the HCP ionization and responses. And so, therefore, a more accurate evaluation of HCP amounts can be performed using an MS labeling strategy like cold, heavy labeled isotope analogs and multiple reaction monitoring. And this can be adopted as a follow-up or an alternative method. So to review the main points, the regulators are interested in seeing evidence that can corroborate HCP ELISA results, and LCMS provides a complementary alternative that is accurate and sensitive with high reproducibility. It's not limited to HCPs that produce an immunogenic response or by manufacturing process constraints. We have highlighted 3 areas of attention when planning an HCP analysis strategy: the spectral library type that determine the depth of HCP analysis, sample selection for quantitation, which is dependent on the level of the screening that might be required, and finally, consideration for the data expectations during the data review process. The strategies and rationale for implementing a particular HCP MS analysis will differ depending on the product, the process, and other process-specific variables, and we hope we have provided some useful insight for when you make these decisions. Thank you for listening, and please see our details for any further questions or feedback after the talk.
Great, thanks. And with that, we're going to start asking some of your questions to our speaker. Okay, are you ready? The first question is, we're using the same cell expression system for different products. Is it possible to reuse the spectral library for each product?
Right, hello. So different growth and stress conditions can have an impact on the protein that's expressed. And so for different products, this might vary, and therefore we would recommend characterizing the cell originator that's been used in the manufacturing process for the specific drug. Again, so if it's different lots of the same product, in general terms, one lysate library might be fine, as we wouldn't normally expect large changes in the HCP profiles from batch to batch. But you would still need to closely monitor for any changes, or for example, a deviation to the extraction process, which might necessitate reanalyzing the host cell lysate and the library. That's answered that.
Okay. If LCMS is not a replacement for ELISA, do I still need to develop product or process-specific ELISA?
This is still a grey area. So as submissions with off-the-shelf ELISA kit data and MS data are only just going in front of the regulators, I suspect the need for a product or process-specific ELISA will depend on the coverage from the commercial kit across the HCPs. So if this is high, then the commercial kit plus the HCP MS data will probably be enough. But if the commercial kit ELISA coverage is low, then yeah, you'll need probably a specific ELISA plus the MS.
And the HCP data analytics sound complex. Do you use a specialist programs?
Yes. So depending on the instrument or system that you use, there is instrument-specific processing software tailored for the setup and the raw file type. We use both Cyax and Water systems, and for each one, there's specific software, namely Progenesis or the SWOS acquisition micro app. So they perform the peak picking analysis and they calculate the areas that are taken forward for the quantitation estimates. These software packages produce large volumes of data, as you can imagine. So depending on the nature of the sample under investigation, we've also, to assist with this process, we've also spent time developing our own in-house data filtering methods. And so along with this initial processing, it helps us to target the real identifications and eliminate further false positives. And as I've mentioned during the talk, we don't like to rely too heavily on the software alone. So we also perform the extra manual reviews to assist in reducing any HCP oversight.
Okay, it looks like we have time for just one more question. If we didn't get to your question, we'll be passing those along to Christina directly. So please go ahead and continue to type in your questions. But the last question for this webcast is, how long does a 2D LC library creation take?
The short answer there is that it's product specific. What a 2D LC method will give you is better separation. So yeah, it can improve the depth of the reference library. It will screen for the very low level HCPs. It's just important to note that it will take more time, so more in the order of weeks to a month, depending on the product. So yeah, you will get that better coverage, but at a cost of time.
Okay, well, thank you, Dr. Morris.
Thank you. Thanks for having me.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.